All right, good morning, everyone. Thank you for sticking around on this last rainy day. It's been a terrific conference. So we're going to start today with a really great session on tumor immune microenvironment and metabolic reprogramming. I think I'm going to ask the audience to maybe hold your questions until all the speakers are done presenting, and we're going to have a panel discussion. So the first up is Dr. Zubeida Cruz Monserrat from Ohio State University, telling us about the link between obesity and pancreatic cancer. Thank you.
Thank you very much. Could you reset the clock to 15 minutes instead of 30? Because I don't think we got 30 minutes. Okay, well, thank you for the introduction and the opportunity from the organizers to speak today. Today, I'm going to tell you a little bit about our story about our association of obesity and how it connects to pancreatic cancer. I have no disclosures besides if you heard my talk on Tuesday, this is not the same talk, and we're going to talk a little bit more and expand on that. So there are known risk factors for pancreatic cancer that increase the risk, and one of them is obesity, which is we consider it a modifiable risk factor. As we know, this is a worldwide problem, including in a problem in the US, where the percent of obese individuals is measured by body mass index in adults has been increasing over the years, where you can see here that in 1990, the percent of obese individuals in the States was, in teal, about 10% of the population in those states. And in 2020, we're in between 30 to 40% of the populations in the States are overweight or obese. This is concerning because now we're recording data of young adults, especially high school students, and this is also increasing. And as we know, there has been some evidence about incidence of cancers at a younger age, and we don't know if this association, if this correlation may actually be a causation. So we're looking at that very carefully, especially all of us that study obesity. So there are about 13 types of cancer that are associated to being overweight or obese. Pancreatic cancer, of course, the one that I'll be focusing on today. So we know pancreatic cancer is a deadly disease, and by 2030, it is thought to be the second leading cause of cancer death. And a lot of these trends, again, are associated with the increased risk in obese individuals in the United States. But again, we don't know causation. And this is where I usually talk about how do we measure obesity. Most people use body mass index. In the world of body composition, that is one not a good measurement, just because you can have a high body mass index and have a lot of lean mass, which is actually good for you. If you're a bodybuilder, you're going to have a high body mass index, but it's an opportunistic measurement because some of the other measurements you have to think about prospectively, especially when you're doing your trials, because they're more mechanistic. And like DEXA imaging's waist to hip ratio is one of the ones that people in the energy balance like to use. But again, body mass index is a way to start, and 30 equal to 30 or more is actually defined as an obese individual. So, how can we, and some of the reasons for this is the contribution of the Western style diet that has, very adopted by many people in the United States and actually around the world, composed of refined sugars, low fibers, proteins, high carbohydrates, saturated fats that lead to this increase in weight, fat, inflammation, and insulin resistance. And so, in terms of what the risk factors are, we really can't study that in humans very well, and that initiation, especially because pancreatic cancer is a rare disease to begin with. So for that, we use models we use the KRAS model, in particular the KC model, where we feed them a Western style diet, and then the mice get obese, either without KRAS or with KRAS mutations. But if you have KRAS mutations, you also gain weight. This is kind of like what their body mass, what their weight is over time. And then if you have a KRAS mutation, you start seeing that even after 30 days of a high fat diet, you start seeing the increased acceleration of these panning lesions that are normally not there in the KC model early on. You actually need that progression. The KRAS alone needs a little bit more time. Now, if you leave the mice on the high fat diet for a really longer time, and when I mention high fat diet, there's different types of high fat diet. So I'm talking about high lard, although these diets were isocaloric for carbohydrates and other components. But in this case, you actually, if you leave them longer, the mice develop tumors, and their survival is actually less compared to mice that are on a regular diet. But as you can see on the top, on the control diet, the mice on the KC mice will develop these lesions over time. But when you add them on the high-fat diet, they're more aggressive. So we asked the question, what could happen if we incorporate weight loss interventions? Could we delay or prevent PDAC development all alone? And the Casey model is a great model for this because it's a long-standing model. It takes a little while for it to develop, and so we set up to do that. And so at the time, this studies actually have taken a long time. It's been 10 years that we've been working on this. Oops, that the glitch came back. So look to my left. Um, so we actually partnered with the community back at when I was working at MD Anderson about 10 years ago. And the community had this program called Power in Motion. And so this is volunteers that came to a 10-week running program, and they've never run before. And so they spend 10 weeks in this tailored running clinic where they taught them how to run, and then at the end of the 10 weeks they will do a 5K. That was like the goal. This is the individuals without cancer, just they are just voluntary. They wanted to learn how to do their first 5K. That was one of the inclusion criteria. And so they measured, they took blood before the intervention and then after intervention, but not the blood after was not after the 5K, but like days after. And then they had DEXA scans and other amphimetric measurements. And it was very interesting. And this is where the waist to hip ratio comes into play, which is the one that the people in the energy balance field like to use. They saw a significant decrease in these patients after the 10-week program. And we were fortunate enough to have access to the blood samples from these patients. And we run a panel of Luminex, and then these were the ones that were significantly decreased. And some of these they're very well known in our field as to be some pro-inflammatory markers that might lead to cancer. But again, these individuals did not have cancer. It was not the goal of the study. And so this is when it was very interesting to see these changes. Can we recapitulate those changes in our mouse models? And so for these, again, we took them obese mice, and we got them obese, and then we either switched their diets to a low-fat diet or to a diet or to increase their physical activity. Physical activity has been associated to decrease the risk of multiple cancers, including pancreatic cancer, in epidemiological studies. And so we took the KC mice again, and we activated KRAS. We put them on the high-fat diet for 4 weeks. We knew that at that time we're going to have lesions. What happens if we combine them with physical activity? And so you ask, how do you put a mice to run? These are like physical activity wheels that they can voluntarily hop on, and they will run, and then they're monitored by via Wi-Fi, and we can see how many revolutions the wheel will have. And then the mice that were not on the intervention had access to a wheel, but the wheel will not run or turn over. They will not have time to run. And so you could see that the mice actually love to run. They actually, this is the equivalent of a 5K in 4 hours. We did it 4 hours a day, 5 days a week. And then the mice did prevent the weight loss from being on a high-fat diet. But as you can see in the gray bar on the bottom, they will delay the gain weight, but they will eventually gain weight. I mean, the body kind of once the exercise activity plateau. But the body weight change was significant in the KRAS mice. And when we saw the histopathology of them, there were less lesions and less pancreas tumors in those. We saw decreased inflammation and decreased fibrosis in these mice, which was very encouraging. And none of the mice in the intervention group actually had any evidence of pancreatic ductal adenocarcinoma compared to the mice that were left on the high-fat diet alone. At this short intervention, it was only a 4-week intervention. Interesting, the pathology scored kind of correlated with the amount of activity during the entire duration of the study which was very exciting. Now, also interestingly, a lot of these cytokines that we saw in the humans were also decreased over time. We measured blood over time, and like IL-6 and lipocalin-2, which you heard me talk before. And so, very briefly, we also look at the at what will happen if we combine the physical activity intervention, which also the dietary intervention, because in the first experiment we actually left them on on the high-fat diet, and so again, these mice will actually love to run, and you can see that both either just the diet change or the diet and the physical activity had significant decrease in body weight, and the combination actually was more powerful in preventing the formation of tumors. Now, when we move into an orthotopic model, this was very interesting because when you get the mice obese, you have them have the intervention for about a month, either the weight loss via diet or diet and exercise. When you actually see a significant decrease in fat mass in both, I mean, the combination was even more powerful. But when you look at and then they significantly lost weight. But when you look at the tumor weight in the end, it was actually not significantly different compared to the mice that were on the high fat diet, that stayed on the high fat diet. And so this kind of says that these weight loss interventions of physical activity appear to be preventive of obesity associated PDAC, but in the treatment setting, they will still need to be combined with some other kind of therapy. And in our study, we actually combined it with gemcitabine. I don't have enough time to tell you about this. And we could see that there was an increase in CD8 positive T cells in the tumors of those mice. And so we set to look at what were some of those inflammatory or anti-inflammatory changes that could happen, or factors that were in the adipose tissue, to kind of inquire about changes in the adipose microenvironment, moving away from the tumor microenvironment a little bit. And some of these, this is just an old Affymetric at the time. I'm going to show you RNA-seq in a little bit, but we saw these anti-inflammatory molecules, one of them being IL-15 receptor in the adipose tissue, but this was bulk adipose tissue. And so during that time, there was a study that came out from in Cancer Cell from Dr. Versage's group, showing that exercise induced the IL-15 axis, and in combination with immunotherapy was actually a very good promoter of anti-tumor. But in these studies, were done a little bit different. Again, they were not done in the context of obesity. They were done in the context of lean mice undergoing an exercise, a little bit more of a forced exercise in a treadmill setting, and in combination for therapy purposes, not for the prevention purposes. And so we set up we partnered with a collaborator at Ohio State that has an adeno-associated virus specifically to the adipose tissue, to the white adipose tissue, to inject IL-15, or have the adipose produce their own IL-15. And we were able to confirm that the IL-15 was increased in the adipose tissue. And interestingly, when we actually do orthotopic tumors, these tumors actually were smaller than those that didn't have the IL-15 intervention. However, when the mice were obese first and they were treated with the IL-15 virus to the adipose tissue and have the the own system produce IL-15 in the adipose tissue as shown here, the tumor weights were actually the same, and there was no significant. So this is where we wonder what other changes could be occurring in the adipose tissue that could be leveraged to delay obesity-associated PDAC, especially when interventions such as exercise interventions might not be feasible for our patients, or in some cases. And so that we went back to our experiments and look at RNA-seq data, and we again validated that a lot of these pro-inflammatory cytokines that are associated with tumors were actually decreased after our weight loss interventions. And so in looking at the adipose microenvironment, it was striking the amount of variability or change after the diet-induced weight loss cell composition in the adipose tissue. More interestingly, we saw like different compositions of the white adipose tissue, and when we like home in into the white adipose tissue, and more thermogenic white adipocytes were a process called beiging adipose tissue. And this is probably my only immunology slide of the morning in my talk. But one of the striking, interesting things too is that a lot of the macrophages were decreased. So the adipose tissue associated with crown-like structures that are surrounded, adipocytes that are starting to die and surrounded by macrophages, we saw a big reduction in the abundance of these macrophages in the mice that have actually lost weight. And interestingly, a decrease in the percentage of myeloid-derived tumor suppressor cells, the MDSCs, in the diet-induced weight loss. So in summary, I've shown you data that inflammation can be decreased after a 10-week intervention in individuals who were obese or overweight that wanted to start an exercise intervention, such as a 5K, and start running. And that when you induce obesity in a high-fat diet, using a high-fat diet, diet-induced obesity model, and put those mice to either increase their physical activity or having a weight loss intervention such as dietary intervention on in combined, you see decrease in inflammatory features, fibrosis and PDAC, and changes in the overall adipose microenvironment. And so we're now working on looking and comparing how these changes in the adipose tissue of mice compared to those in the human disease. And with that, hopefully we can talk more about this and ask any questions after the session. And I'd like to thank my lab and a lot of the collaborators and funding that have funded this and many other projects in the lab. Thank you very much.
Thank you for this great talk. We're going to transition to the next one by Dr. Mingyang Liu from Peking Union Medical College, telling us about the crosstalk of tumor and immune microenvironment in pancreatic cancer progression.
Thank you for the introduction. Dr. Liu cannot be here today because of unforeseeable reasons, and she really apologizes for that. I'm Dr. Zijun Zhou from the University of Oklahoma Health Campus, and I will represent Dr. Liu to present. Pancreatic cancer is the third leading cause of cancer-related death in the States, and unfortunately, only 30% of patients can receive surgical resection, and most of them are resistant to chemoimmunotherapy, and the 5-year survival is only 13%. So I don't think I can emphasize too much on the importance of tumor microenvironments in pancreatic cancer. So Dr. Liu's lab mainly focusing on how tumor microenvironments drives cancer cachexia and chemoresistance in pancreatic cancer. So why we study pancreatic cachexia? This is a syndrome of a systematic wasting, and over 17 to 80% of patients will develop cancer cachexia, and the clinical symptoms include body weight loss, muscle wasting, decrease of appetite, etc. So our lab is the first to identify the role of tumor in the microenvironment in driving pancreatic cancer cachexia, and we show that the macrophage in tumor microenvironment is associated with the cachexia potential of different tumor types, and we show that pancreatic cancer patients with higher percentage of macrophage have worse prognosis, and is the driving force of cancer cachexia. And mechanistic study shows that cancer cells secreted CCL2, which recruit and activates macrophages, and then promotes the secretion of CCL5, which will activate the TROP6 NF-kappa B signal pathways to upregulate TWIC. And then TWIC will target the FM14 and the RAL-B signaling axis to induce muscle wasting. The other direction is chemoresistance. We all know that chemoresistance is associated with the immune suppression in pancreatic cancer patients. So a recent publication shows that a crosstalk between tumor cells, macrophages, tumor cells, neutrophils, and T cells that drives the chemoimmunoresistance. First of all, we established the chemoresistant cell lines, the gemcitabine resistant cell lines. we show that when we co-culture the T cells with these cell lines, we show that it will decrease T cell migrations. And through RNA sequencing and screening, we identified ZEP1 as a key transcription factors. We further use immune-competent and immune-deficient mice to establish the orthotopic mouse models. We saw that in the immune-competent mice, where we knocked out ZEP1, the anti-tumor effect is more potent than the immune-deficient mice, indicating that the immune cells play some role here. So next, we combined ZEP1 knockdown with gemcitabine, and we saw that it will increase the sensitivity of gemcitabine and also extend the survival of mice. Later, we did the single cell analysis, and we showed that when we knock down ZEP1, it will increase the percentage of total CD8 positive T cells and the cytotoxic T cells. And next, we did the subtype analysis of different CD8 positive T cells. We identified the GZMA positive effector T cells that are dramatically increased after we knock down ZEP1. And we further conducted the spatial transcriptome analysis and showed that when we knock down ZEP1, it will increase the infiltration of these cytotoxic T cells and also increase the cell-cell interaction analysis show that knockdown ZEP1 will increase the interaction of these T cells, these subtype of T cells with the tumor cells. And next, we show that whether knockdown ZEP1 will influence the activity of these T cells. We saw that when we knock down ZEP1, it will increase these biomarkers associated with T cell activations. And also, we established the CAR T immunotherapy using human cells and mouse cells. We saw that knockdown ZEP1 will have synergistic effect with CAR T immunotherapies in both in vitro and in mouse models. And our previous study shows that ENT1 is a key transporter for gemcitabine. Here we show that when we knock down ZEP1, it will increase the expression of ENT1. It will increase the expression of ENT1, and then we confirm using the diagram using the diagram to show that when we knock down ZEP1, it will increase the uptake of gemcitabine by the pancreatic cancer cells. And since pyroptosis is also important, we also show that when we knock down ZEP1, it will increase the pyroptosis in these cell lines. So next, we know that there are multiple immune cells in the tumor microenvironment. Besides T cells, we also analyze other cell types, and we saw that when we knock down ZEP1, it will increase the interaction between tumor cells with neutrophils. So we established a co-culture systems using KBC tumor cell lines and T cells and neutrophils, and then we used the sorting to sort these cells, and then did the qPCR to analyze the markers associated with T cell activation and neutrophil activation. And we saw that when we co-cultured with the when we co-culture the T cells with the neutrophils, it will decrease the infiltration and the activity of CD8 positive T cells. And when we knock down ZEP1, it will increase the activity of anti-tumor immunity. And in the mouse models, when we combined gemcitabine, ZEP1 knockdown, and neutrophil inhibition, it shows more dramatic anti-tumor effect. And next, we wonder what's the underlying mechanism that drives the interaction of the crosstalk between the tumor cells and the CD8+ T cells. We did also did a single cell analysis, and we identified the CXCL16 signal pathway as a key regulator. We saw that when we knock down CXCL16, it will decrease the migration of CD8+ T cells and its anti-tumor activity. And when we combine the CAR-T immunotherapy with the CXCL16 knockdown, it will increase the activity, anti-tumor activity. And next, we wonder we wonder why the CXCL16 was regulated in the tumor cells, and we did the cut and tag qPCR axis. We saw that the acetylation of histone of the CXCL16 promoter decreased, indicating a epigenetic modification during this process. And when we knock down Z1, it will decrease the level of CXCL16 in the tumor cells. And next, we combine the gemcitabine with a HDAC inhibitor and immunotherapy. We showed that the combination will induce synergistic anti-tumor immunity in mouse models and increase the infiltration of CD8+ T cells and decrease the level of neutrophils in tumor microenvironment. And we also use the CAR-T therapies in vitro, so that when we combine the HDAC inhibitor with CAR-T, it will decrease the anti-tumor immunity of CAR-T cells. And then we also validated these findings in mouse models and have similar findings. And lastly, we compare patients with chemo-resistant and chemo-sensitive, and we found that in patients with chemo-resistant tumors, the Z1 expression increased and CXCL16 levels and CD8+ T cells decreased in the tumor tissues. And when we separated these patients based on CXCL16 expression in tumor tissues into low and high groups, we saw that those with chemo resistance lower expressions in the tumor tissues, and CXCL16 low is associated with worse treatment outcomes of these patients. So in summary, this study identified how Z1 sensitized pancreatic and how Z1 induced the chemo resistance in pancreatic cancer patients, and we identify a interplay between the tumor cells with neutrophils and T cells that drives cancer chemo immunoresistance which involved epigenetic modifications of CXCL16 signaling pathways. We also have other studies showing that tumor-derived microRNA 2038 can promote apoptosis of muscle cells through the Z1 signaling pathways. And this is Dr. Liu's group. And with that, I would like to thank our collaborators at OU and PUMC and many other collaborators across the nation. Thank you very much.
Yes, perfect. Thank you. All right, thank you for a great talk. So next one is by Dr. Histaka Kobayashi from Kawasaki Medical School, telling us about photoimmunotherapy of pancreatic cancer. Very intriguing.
Thank you very much. Good morning. Thank you very much for inviting this nice meeting. I like to focus on the near-infrared photoimmunotherapy. This is actually the new therapy, cancer therapy to develop for the any cancer. And also that potentially that can be manipulate the tumor microenvironment that I want to talk today. So this my talk will be start with the what is this technology, why that work, and then we have already in the clinical approval and then treated the head and neck cancer patients. So I'm going to touch on the clinical results. Then I like to move on to the pancreatic cancer for manipulating the tumor microenvironment with this technology. So three major cancer therapy is actually the surgery and the radiation, the chemotherapy. You try to minimize the cancer cells. That's a goal, but then that's also damaging on the normal cell and then also including the immune cell. But so the idea of therapy is, I think the cancer cell should be minimized, the number of the cancer cells should be minimized, but then enhance the immune cell. So this is, if we can do both at the defense and offense at the same time, that should be a best, actually that's a best MVP award. So like the Shohei Ohtani in the Major League Baseball. So it's a tumor microenvironment. Is actually the mixture of the cell. The tumor is not consist from only from the cancer cell. So unfavorable cell is the cancer cell, and then cancer stem cell, and then immunosuppressor cell, and also that especially in the pancreas cancer, that's a tumor infiltrating fibroblast or cancer-associated fibroblast. That's also the hamper for the pancreatic cancer therapy. But then still there is a supportive cell, is the immune cell, and then tumor B cell that's going to be trafficking the cell, and also tissue stem cells still remains. So that should be work for repairing the after the cancer cell is removed. Then photoimmunotherapy is actually like this. So we use the antibody to selecting the cancer cell, and then we put the photosensitizer on it to activate the cytotoxicity. This way is actually only the antibody bound cell is crushed by ignited by the light. And then, so this is what I show you. The so target cell is a plus, minus is a non-target cell. We co-culture is side by side, and then put the antibody on, and then expose the light. If so, at that time, only the antigen positive cell is swollen and then burst within a couple minutes. And then non-target cancer cell is totally healthy. So we can selectively, self-selectively, deplete or crush the target cell, whatever we like. So this is actually the mechanism. So antibody bind to the surface receptor, ignite by the light, and then activated the photosensitizer. So cell membrane damage, the water is rushed in, and then cell is swollen, and then some big tear on the cell membrane, and then the contents going to be comes out. So this is another way to show, this is actually try to visualize the cell membrane by the phase contrast microscope. So this is only 1 minute video. So it's a not accelerated video. So tumor cell is actually swollen 2.5 times as much of the volume within a minute and then burst. So it's a very quick process to kill the cell. And then this biologically, so this is usually calling the immunogenic cell death. So cell is swollen and then burst, and then contents going to be comes out. So, but then also that the dying signal, it will be activate the immature DC, and then the DC going to be maturated, and then burst cancer cell release the all neoantigen of the cancer, like the the mutated RAS or whatever. So that's going to be recognized by the maturated DC, and then the maturated DC going to be educate the naive T, and then naive T going to be primed, proliferated, and the new set of that CD8 going to be produced. And then this CD8 going to be taking care of the survived cancer cell. We can crush the 90% of the cancer cell by the light, but so only 10% is still survived. So this cell, this CD8 can be taking care of the leftover cancer cell. And then we start the head and cancer clinical trial in 2015, so already 10 years ago. This is a phase 1 trial, but still the clinical, exactly we are doing the same way. So we drip infusion of the antibody conjugate into the patient for 2 hours the day before we expose the light, and then we expose the light. So phase 1 study, we did the dose escalation, but then this is actually the rapid result from the phase 1 in the Rush University, Dr. Stenson in the Rush University, written in the newsletter. So almost immediately you can see the tumor start dying and then turn white and melt away. Destroying the cancer cell causes almost no damage to the surrounding tissue and then very safe from the systemic perspective. So acute change is like this. So this patient have recurrence of the tumor in the back of the throat. And when we expose the light, immediately after, as she mentioned, so color turns white, and then some of the blood will be on the tissue, and the next day, that tissue is melted away and fallen down. So this is a very quick process. And this is a little bit subacute process. So patients have big subcutaneous recurrence of the head and the tumor. So this light only penetrates 1.5 centimeters. So we can insert the needle. This is actually the 20 gauge elastic tube into the tumor, and then we're going to insert the light fiber inside of the syringe, and then expose the light from the inside of the tumor. So we can do the 5 exposure at once. So even 10 needle, we can do just twice to expose the light every 5 minutes. So 1 month later, so almost a big tumor is melted away and then gone, but still have some erosion on the surface of the skin. But 2 months later, so that the skin is back and then hair is grow back. So because this tissue stem cell on the skin is still survived, and then cancer cell by itself is removed. So that's the reason why wound healing is very great. And then this trial is, this study is already approved in Japan in 2025 years ago. And then this cost for $60K, but still for insurance coverage, so patient only pay for $2 to $3K after the insurance coverage. And then 2024, so last year, end of the last year, we treated over 400 patients with over 700 times. And then this year, we also treated 300 more patients and then 500 times of the therapy. So now it's the number of the patients is 700 patients in the 1200 therapy have already been done in the 180 plus hospital in Japan. So have a lot of clinical results here. And then already the clinical result is appeared on the paper. So overall response rate is really great. So 89% and the DCR 100%. So that means most of the tumor can be shrinking down. But and then the very good thing is actually that the second from the last line. So this have been presented in the ESMO in the last month. So among 25 recurrent epipharyngeal cancer patients, 21 patients got CR. And then another 3 patients is a good PR. So 96% of the recurrent epipharyngeal cancer patient treated with this therapy is, the very good outcome of the after that. Go back. Yeah, this is one of the patients of the epipharyngeal recurrence patient. So left side of the picture of the endoscopic picture shows the ugly tumor on the top of the throat. But after 1 month, a couple weeks after this side of the tumor is gone, but still have a little bit ulcer there, but biopsy is negative. And then this is a PET scan, FDG. The day before, this patient have a very good uptake of the FDG, but then the day after exposure of the lights, all the tumor tissue cell is losing the glucose uptake. So that means the glucose metabolism totally stopped, even during the exposure of the light. So this is 98% of the activity of the FDG is gone in a day. And then this can do under the endoscope. So endoscope, so it's a the light fiber is very thin, so we can go through the endoscope and then even stick into the tissue through the endoscope. So I think it's a pancreatic cancer. If so, at that time we can go through the ERCP technology, or even use the EUS from the stomach to puncture through the stomach wall to get the pancreas. That might be one of the good way to reach to the pancreatic cancer. And then, so move to the tumor microenvironment, we can crush not only the cancer cell, we can crush any cell we want to target. So this study, so we're gonna target the Treg. So Treg is one of the cell that my very long time good friend Shimon Sakaguchi got the Nobel Prize in the last month. So this cell is expressing a lot of CD25. So we can use the CD25 antibody and then use this technology to crush the Treg in local. That means the tumor tissue and probably at regional lymph nodes. That's only that the local area, because the Treg have a very big role on the suppressing the immunity. So that's the reason why if we're going to deplete all the Treg from the body, at that time, we're going to get the very severe autoimmune reaction in any part of the body. So it's very hard to use for the cancer therapy. So it's a local, it's really safe. So once we're gonna, once we're gonna to get the two tumor in immunocompetent mouse, and only depletes one side of the tumor, depletes the Treg from the one side of the tumor, that's the top side of the tumor. So treated side of the tumor is disappeared in a day or two, but the untreated tumor is also disappeared in a day 2 or 3. So that means once we're going to activate the host immunity by depleting the Treg, that's effect is systemic. And then, so we sent this to the science on the 2015, so that's 10 years ago. At that time, the reviewer say, this mouse is very small, so light can be penetrated through. So we grow 4 tumor in the mouse, and then very limited to expose to the only one tumor. But still, all the tumor is decreasing and disappears in a couple of days. So this can be really that happened in the systemic improved systemic host immunity. So this method is already approved to the human clinical trial from the 2022, and then now it's still ongoing. So final home of the PIT is actually we're going to crush the tumor, decrease the number of the tumor, and then activate the immunity, and then try to activate more of this harvested T cell to get to the local tumor, and also that survives the T cell for the memory to suppress the recurrence, and also that circulating all over the body to make the cure the systemic metastasis. And then really that happened, and then only the single therapy, that means the crush the tumor or crush the Treg at that time, we can only cure 1 of 10 mouse. But if we combine at that time, single therapy can cure 9 of 14 mouse. So that means the 60% of the mouse can be cured. And then once the cured at that time, that the mouse received again or the same tumor at that time, that tumor never grow back again in the once treated mouse. But of course, that a normal mouse, that the tumor grow and then kill the mouse, all the mouse. So that's this therapy, once cured at that time, mouse can have a tumor, tumor immunity. And then now we are a lot of antibody production in the pipeline, especially in the pancreatic cancer. EGFR is actually positive in cancer, pancreatic cancer, and also PD-L1 and the positive. And then, so we are also focusing on the anthraxtoxin receptor 2. This is actually a very good marker for the targeting pancreatic cancer stem cell. And also the FAP, that means the fibroblast activation protein that's expressing in the cancer-associated fibroblast, especially in the desmoplastic pancreatic cancer at that time, that might be variable. So we are these two on the pipeline. Actually, we are targeting anthraxtoxin 2 receptor expressing cell at that time. That works like the treatment of the cancer stem cell. So that means this cell is depleted from the tumor at that time. The tumor is regressed spontaneously. So this is actually an experiment what we did for this. And then this is the last slide for the summary. We like to deplete, especially for the pancreatic cancer, we like to deplete as much as tumor cell and the cancer stem-like cell, and also manipulate the tumor microenvironment as favorable as possible. So I think that's the way to cure the pancreatic cancer. So that's actually the way we are thinking about. And also we try to improve the method by itself. So now we are using an IR702 HKT. This is a series of the dye superior to the IR700, the current original method. So we have the the free patent dye to move forward. So we're gonna have the meeting this year, next year. This is the first meeting of the photoimmunotherapy will be placed in the NIH. And then this is a worldwide collaboration. Rakuten Medical Incorporation, that is actually selling this drug, is located in San Diego here. So I visited the last month. And also a lot of collaborator is working on. And then this is a partial list of my good collaborator. And then I'd like to thank to everybody. Thank you very much for your attention.
Thank you for a very stimulating talk. So last but not least, Dr. David DiNardo from WashU telling us about targeting the immune microenvironment in PDAC.
Good morning, and thank you to the organizers for inviting me to give this talk. These are my disclosures. I think as you already heard very eloquently from David Tuason, pancreatic cancer, at least the primary site, is a highly desmoplastic disease. This involves high levels of extracellular matrix, as shown on the left, as well as various subtypes of pancreatic cancer fibroblasts. Now, more than this being a barrier to disease, as previously proposed, but maybe debated, you have to recognize that these cancers grow up in this tumor microenvironment and are adapted to them. So the more than being a barrier to therapy, we might think of this as a vulnerability. And I'll give you a story today. I'm an immunologist, a tumor immunologist, and T cells exist in these pancreatic cancers in spite of that fibrotic immune microenvironment. One picture to try to convince you. Up to 30% of primary pancreatic cancers have T cells, and T cells predict survival. We simply haven't been able to leverage the immune system for FDA approvals to treat this disease. Now, I've given that speech before, and several years ago, a very talented MD PhD student, now a physician scientist in the radiation oncology department, said, it's fine that maybe we can't deliver as much drug as we'd like to in this fibrotic environment, and it's fine that T cells don't work, but that doesn't explain why radiation therapy fails to give as much efficacy as needed in pancreatic cancer. It's often given as palliative care after chemotherapy as consolidation therapy at our institution. She said, we can deliver radiation therapy. It's not an issue. We can give high amounts of DNA damaging agents to this tumor microenvironment. And if I read the textbook for radiation oncology, when we give radiation therapy and we kill tumor cells, these dead and dying tumor cells should prime an immune response that then helps prevent metastatic disease. And this is debated in the field in terms of clinically, whether the use of radiation therapy should be done before surgery. So to try to understand this, she did what every talented MD PhD student is. She went first to the human, and she used a couple different cohorts of patients treated with chemo and radiation therapy, or SPRT, and asked whether there are more T cells. And the answer in the simplistic format in these patients was after therapy, she didn't see an augmentation of T cells as shown on the left for CD8 T cells. But it's not that the tumor microenvironment doesn't change. There was a significant augmentation of the collagen density and the fibrosis in these tumor microenvironments. As any surgeon could probably tell you, they hate resecting patients after a long period after radiation therapy. And if we look at this in a more modern schema, as we've done with Matty Rest, another MD PhD student, this is after FOLFIRINOX, and after FOLFIRINOX plus short course SBRT, actually T cells are actually diminished by these therapies in our patient cohorts. But these are patient cohorts, they're selected, they got these therapies for given reasons that aren't for us to study necessarily, and we cannot study the kinetics of disease. So this is a place where mouse models actually can really shine. We use the classic KPC mouse model with the only tweak in it being that we have a neoantigen cassette that expresses a neoantigen, and then we can track neoantigen-specific T cells. And Varintra asked a very simple question: if I give radiation therapy, do I improve the T cell response in these animals? As expected, we give radiation therapy here, a short course of radiation therapy mimicking SBRT. We do limit disease progression over the short term and induce cell death, but long-term survival is not improved. And she didn't see an augmentation of T cell responses, either in the primary tumor or in antigen-specific T cells in the draining lymph node. This is not a mouse modeling problem. Sarcoma and lung models actually show that radiation therapy can prime new T cell responses in our hands in littermate mice. So this suggests in two different cancer systems, in sarcoma and lung, we get radiation therapy can prime a T cell response, but that doesn't happen in pancreas. So what's different? The biggest difference between these mouse models and also between some of these human diseases is the extent of desmoplasia and collagen density in the animal models for pancreas. So Varintra, being a very great student, wanted to study how the tumor cells died post-radiation therapy, and if they died in the presence of desmoplasia. She both mixed fibroblasts and collagen density into standard organoid systems. As shown on the left, if you take an organoid from pancreas cancer, it's responsive to radiation therapy. The pink line on the bottom will show you can ablate these cultures. However, if you do this in the presence of either collagen density or fibroblasts as shown in the figure, you actually limit the efficacy of radiation therapy in these organoid models. And when you combine both collagen and fibroblasts, you actually limit the efficacy even further. The blue bar shows very little efficacy comparably to a single dose of radiation therapy. But we're immunologists, so it's not just important that the cells die, but how they die. As you would know from the literature, after radiation therapy, even before cell death, interferon gets released downstream of DNA damage, and this certainly still happens in pancreas cancer and pancreas cancer organoids in the gray bars. Interferon is released after radiation therapy. But if you do this in the presence of high-density collagen, you limit the release of immunotherapy, release of interferon, sorry. And this is true across a whole host of interferon-related genes and pathways and cytokines. This suggests that desmoplastic response both supports the cell survival, but also supports in the short term the immunologic way they die, dampens it. Okay, we've studied the impact of extracellular matrix on cancer cells before, and one of the pathways we choose to look at a lot is focal adhesion kinase. Downstream is stiff extracellular matrix. There's activation of integrins that activates FAC. FAC itself then in turn can regulate survival pathways such as YAP-TAZ or AKT, invasion, and cytokine production. Now I'd be dismiss if not saying that FAC actually activates also cancer-associated fibroblasts towards a myofibroblast phenotype. And I'm going to show you clinical translational data on both these compartments in a combined trial in a minute, but I'll go through the remaining preclinical data. So not showing you the in vitro data, but straight going in vivo, our hypothesis was a FAC inhibitor to disrupt the tumor intrinsic extracellular matrix signaling would actually augment interferon. We saw that in vitro. We can see this in vivo. Mice treated with both RT and a FAC inhibitor systemically show an augmentation of interferon in the tumor cells. In this image, I'm showing you that with phospho-STAT1 signaling, but also in the surrounding tumor microenvironmental cells, such as CAVs, which upregulate interferon in response to dual therapy, but neither single therapy alone. As summarized in the paper, this results in a better T cell response, including immune priming. And in these preclinical models, we could observe the augmenting radiation therapy with a FAC inhibitor leads to disease regression, even in recalcitrant genetically engineered mouse models, as shown on the top right. And this is better than just adding gemcitabine on top of radiation therapy. Interestingly, that initial tumor regression is totally T cell independent. However, the long-term disease stabilization, once tumors regress, was dependent on T cells, as shown on the bottom right. So in summarizing these sort of preclinical data, so I can show you some of the clinical data, these suggest that when we inhibit FAC inhibition and block that stromal signaling to the cancer cells, this augments the type of immunologic cell death present downstream of radiation therapy. Type 1 interferons are released, and this allows for immune priming, as we showed in the preclinical models. But mice lie. So does this happen in human? It's an important question. Fortunately, at WashU, we have very talented radiation and medical oncologists. Hyun Kim and Patrick Gearson ran this trial. It's a 5-fraction SPRT with a FAK inhibitor given to up to 1 year. And we did pre-post-treatment biopsies. So this trial is actually complete, fully enrolled to 36 patients, and they will present the data at ASCO GI. We are excited about the responses that we get. But this is the RAS inhibitor era, and so we're looking at how we can build on this, and I'll show you a little bit of that. I'm only going to present the biomarker results. So in the first 6 patients as we published, I'm going to show you 24 patients in a minute. That mechanism of action that our radiation therapy plus FAK inhibitor augmented interferon responses was retained. You can see this in the single-cell RNA-seq analysis from pancreatic cancer cells, but also the tumor microenvironmental cells, tumor-associated macrophages and CAVs, all have augmented interferon responses. In these first 6 patients, we also saw an augmentation of the number of T cells in the tumor microenvironment in the biopsies, and the T cells had better effector function. I'm going to tell you that interferon response is held across 32 patients in single-cell analysis I showed you, but the T cell responses are more varied across patients and are linked to differential tumor intrinsic biology, which I won't get into. So we've been fortunate enough to collect tissue from 33 of these patients, including an SBRT alone arm and an SBRT defatinib arm or FAK inhibitor arm, and we have 78 samples and greater than 500,000 cells in a single-cell object. Our questions that I'll propose today are around how this changes the tumor microenvironmental cells. I'll show you some CAF data. 6 minutes, I should go slower. And how this changes the tumor intrinsic biology across these patient samples. First, the mouse models had suggested to us when we give a FAK inhibitor that we decrease the amount of myofibroblasts. But this is in mice, which then establish fibrosis over time. In patients where fibrosis exists at the augmentation of therapy, we were interested in whether a FAK inhibitor in the context of SBRT would alter myofibroblast presence. So this is single-cell RNA-seq. As you can see on the left, there's myofibroblasts, there's inflammatory fibroblasts, and a couple other subsets of CAVs. When you guess SBRT, as we would expect from histology, there's an augmentation of the myofibroblast phenotype in the CAVs that are present there, actually more CAVs on number. So shown in the middle with the high red population. On the right, though, when we give defatinib, the FAK inhibitor, we see that we don't see this augmentation of myofibroblasts in the context of SBRT, but actually an augmentation of inflammatory CAVs. And we're exploring what this means for cell signaling. The other thing we can do is we can splay patients. We know the patient outcomes by good and bad outcomes, or more average PFS fitting with our institutional controls, and a good PFS, those that are living with progression-free survival 9 months or greater. Sorry to use these terms, but I'm waiting for Hyun to show the clinical results so I can quantitate it here. When we look at those patients who do better than average, we see that our mechanisms of action, which is RT-FAK in tumor cells releasing interferon, are actually upregulated further. What I mean is, in nearly all patients, we see upregulation of interferon signatures in the tumor cells, but they're upregulated to a greater extent, and antigen presentation comes up in those patients who do better. This is important because we're treating localized disease. The majority of these progressions are at the metastatic site, suggesting we need to mount a better T cell response, and the first step might be immunologic cell death. But we also can look at mechanisms of resistance. In those patients who do not as well on this trial, we see augmentation of the RAS-MAP kinase pathway ending in MYC activation. This latter point is something we can do something about, which is actionable. 4 minutes. We've seen upregulation of ERK and MAP kinase pathways in mouse models and in other clinical trials using FAK inhibitors. This is known. This has allowed us to combine a RAF-MEK inhibitor, avutometinib, in combination with FAK inhibitor and DNA damaging agents to work at both the stromal compartment and the tumor intrinsic compartment in concert and overcome these therapies. We've done this in metastatic models, as shown on the top left and published in 2024. A combination of a RAF-MEK inhibitor plus chemotherapy augments tumor regression in this metastatic model. And this was translated with help from PanCAN into a multi-institutional study of Gem-Abraxane, defatinib, and avutometinib. On the bottom, following this radiation therapy strategy, we put a RAF-MEK inhibitor, a FAK inhibitor, and radiation therapy in animal models. Well, FAK plus radiation still gives us tumor regression, even this recalcitrant model. We get greater tumor regression and greater control with the full combination. And this is a trial we're hoping to open up in the next month to enrollment, again with Hyun Kim and Patrick Gearson. Mechanism of action, since I've been a little bit skimpy on that to date, is that I've shown you that extracellular matrix is very dense in pancreatic tumor microenvironment. This signals cell intrinsically through FAC and other mechanisms. FAC-specific mechanisms can augment survival through AKT and actually drive MEK expression. When you give a RAF-MEK inhibitor, you downregulate the RAF-MEK complex, but this is not lasting. ERK and MYC get reactivated, and they sometimes get reactivated through both that extracellular matrix pathway, but also through growth factors such as FBF or EGFs produced by the cancer-associated fibroblasts. We hope with FAC inhibition, we're hitting both that cancer-associated fibroblast pathway as well as this tumor intrinsic pathway and can leverage this. Okay, lots and lots of words said very, very fast. What is the big take home? I'd like to tell you that I think the tumor microenvironment, rather than being a barrier to therapy, may be a vulnerability. We can modulate that tumor microenvironment that pancreatic cancer cells for a moment may not be ready for that. Now they will adapt and activate other pathways such as RAF-MEK. And those appear to be additional vulnerabilities which we can target, and this can be tractable. Specifically here, collagen, extracellular matrix, and FAC signaling may limit the immune stimulatory action of DNA damaging agents such as chemotherapy, but I use radiation therapy as an example here. And we think that's important and tractable. I'm a PhD scientist who's running a laboratory. This is clearly team science. This is not specific to this particular talk, but we have a large group of radiation oncologists, medical oncologists, and surgical oncologists, pathologists we work with to be able to do these types of studies in the context of our SPORE and in the context of other clinical trials. And then the most important people on this slide are actually at the bottom right, our brave patients who volunteer to go on to these studies, let us take tissue and try to discover the biology at play when we're giving them a new therapeutic agent. We don't know whether it's going to help them or not. And with that, I'll stop.
Yes, thank you so much to all the speakers, and they kept us so on time that we have enough time for questions, about 10 or 15 minutes worth. So please come on to the microphones.
Hi, Dr. DiNardo, or Mark Wiley from UCSD. I had a question for Dr. DiNardo. I thought it was a really interesting talk. I was curious if you saw any evidence of changes in the TGF-beta signaling pathway in some of your data, just with some of the overlap with like MAP kinase and things like that. I was curious if you saw anything there.
Can you hear me? Yes, TGF-beta is downregulated by the FAC inhibitor in the context of both the mouse models and the clinical samples in the fibroblasts. I'm sure we know the answer for the pancreatic cancer cells, but I can't tell you that I know for sure. I will also share that there is a shift towards the basal phenotype, dramatic by the SBRT alone, and still happens in the presence of SBRT in the FAC inhibitor, suggesting that TGF-beta may be higher in them, but I don't know the precise answer.
Great, thank you very much.
Hello, so this is Marie Hasselund from Columbia University. I also have a question for David DiNardo, unfortunately. I was wondering, you were showing this compelling single-cell RNA-seq data with the depletion of the MyCAFs, and I was wondering if you see also an increase in the ICAFs, and that would somehow suggest that there is maybe more inflammatory signaling going on, if you have any ideas whether that would be detrimental for the patients.
Yeah, good question. We use single-cell RNA-seq in the context of, in the mouse model, we do see this and we can quantitate it. There's a decrease in myofibroblasts on total, CAVs on total, and inflammatory fibroblasts as a fraction goes up. In the patients, we use EOS-guided biopsies, so no histology, but we can use single-cell. We do see a shifting in the fraction that are myofibroblasts compared to ICAFs, and augmentation of the ICAFs. In the short term, we hope that gives us an immune stimulatory possibility. But you're right, there could be a significant deleterious effect of that CAF population by different mechanisms.
Okay, thank you. Minoti update from Sydney. Really enjoyed all the talks. I guess this is a general question, but when you have myofibroblasts cancer cell crosstalk, one of the effects of the stroma is that it can increase the stemness of cancer cells. And I was wondering if any of the interventions that we've heard about today, in an adaptive process, do you get increase in stemness? So in terms of recurrence in these patients that you get a good response about. would there be an effect of that? Have you seen any of that in your mouse models?
Anyone want to comment?
I can't answer, but I'd rather defer.
Has
Okay.
anyone other than David looked at the stem cell markers?
I have not.
Okay. Our hope was with the FAC inhibitor that we were, this FAC inhibitor was, Veristem was a stem cell targeted company like a decade and a half ago. That's because FAC inhibitor can decrease EMT profiles. Our hope was that might at least slow that process. We still see a shift towards the basal phenotype, and including things that we might think about more stem-like or more dedifferentiated in the presence of these therapies in people. But with SBRT and those therapies, so even when they're effective, your resistant cells will still shift towards stemness. So I don't think this is something we can prevent even when we're trying to tractably target it. That's my answer.
Thank you so much for all talk. They were amazing. And I just wonder, have you ever applied the infrared therapy with the microagent to the pancreatic cancer patient? And what were the histological type of the previously treated patients for oropharyngeal cancer? Thank you so much.
Sorry, one more time. Could you?
I just wonder, like, have you ever applied the therapy to the pancreatic cancer patients?
Oh, no, actually, currently just only the experimental. So we actually, have an already approved drug for the EGFR, and then, but the EGFR is not the, it's a perfect marker for the pancreatic cancer. Still, 15-20% of the patients expressing EGFR, so it's possible. But then, so that's the reason why we need the many drugs. But drug development is really costly. So that's the reason why I try to minimize the number of the drug to cover the pancreatic cancer. That's the goal. That's the reason why we are starting for the CAF target one. So I think that can be the decrease the desmoplasticity and also the increase the host immunity. So that can be a positive side. So several different tactics from the other cancers we need for pancreatic cancer. So, but still, that's all experimental.
Yeah, but for the pancreatic cancer, probably it's required the laparotomy for the patients, right?
Yeah, right.
Thank you.
Bhumil from Stanford. Beautiful talks, and I have question for Joveda. So there's a breast cancer studies, obesity related the cancer development, and what they found is interestingly, instead of finding the factors from the obese fat that drive the cancer, they found some factors from the lean fat protect against the cancer. So have you looked at the lean fat?
Yes. So in the mouse models, we're looking at both the lean mice versus obese mice, and then obese mice that went on interventions, either on exercise or exercise and diet. But what I have shown you is just a single cell of the ones that are undergone an intervention. Originally, when we saw changes in the first array that we did where we found the IL-15, those were upregulated in the mice that were obese. So the short answer is we have not dived into things that are upregulated in the lean mice just yet, because we're looking at the changes related to exercise or just obese management interventions. But we will, we're exploring all options, of course. Just got to start somewhere, because we were mostly thinking, there's two ways of thinking. One is, what is it that does, increase the risk? And the other one is what things we can use to treat. Thank you for the question.
All right, I'm going to ask a couple just to round us off. So Zobeda, I'm curious, do you see responsiveness to equal extent in KC and let's say KPC models? Because I saw your orthotopic was not as responsive and I was curious if that was more KPC driven, P53 driven phenotype.
So the obesity also increased the incidence of in the KPC mice, but we don't use have not used the KPCs for our interventions to be able to pursue that question, but it does accelerate. If you can imagine, like the KPC already like lives a shorter time compared to KC, you put them on a high fat diet, it does decrease their mortality, increase their mortality as well.
And I had a question for Dr. Zhao. So you had a very interesting phenotype of gemcitabine sensitivity, and I was curious if you looked at how that correlates with PDAC subtypes, classical basal, with regards to your treatments.
Yeah, that's a great question. We know that currently there are two major subtypes, the basal line and the classical one. I think the basal one are more resistant, but we haven't done anything related to the subtypes.
Okay, thank you. All right, let's thank all the speakers again for a terrific session. Yeah.
Thank you very much for inviting me. So, my old patient, the prerogative to me. Well, not that I ever had any trouble before, but now I have a better excuse, you see. Right? Yeah, the problem was when I was younger. But now I have an excuse. Grumpy old man. Did you ever make me? Well.
Oh, you mean rather here? Yeah, there, yeah. Great. Good tip. Thanks. Yeah, I just want to get right. I've got it here. Yeah, I just wanted to bring up this. Get it right. And I think I've memorized Ali Uj, but I want to make sure I get Robert. All right, let's get started, folks. We've got very interesting session with 4 very eminent speakers. It's my pleasure to introduce the first speaker, Professor Robert Sutton from University of Liverpool. Okay. Well, but that gives us time. Dr. Sutton's one of the all-time pioneers of pancreatic surgery and very critical thinker. They're taking a minute just to get your slides up, but I'll let you, come on up and we'll let you get settled in, okay? Yeah. Any relation to Willie Sutton?
I don't know. I mean, maybe there's a genetic connection, but I'm not aware of a social one.
Do they know this story?
No. Would you like to tell it?
Well, you need to know Willie Sutton. And your slides are up. Okay, go ahead.
Okay.
Won't waste
thank
your
you.
time.
No, that's not mine.
Not?
No.
Oh, okay then. So, Willie Sutton was a very famous bank robber in the 1920s, and when asked, why do you rob banks? He said, that's because that's where the money is. And I think Professor Sutton has taken that's where the discoveries in science and innovations are. Anyway.
Well, I can assure you there's no robbery involved and there's no crookedness. So thank you so much, Martin and Phil. Thank you so much, Melina. Thank you to the APA for this great opportunity to highlight something that may be moving forward from the bench to the bedside. I'm sorry, I've got a lot of disclosures because I really firmly believe that we need to work with industry. But I don't get any money for it, so don't worry. So the global burden of acute pancreatitis is widely known, and the features here listed on the slide are important features, but perhaps most important, we have no drug. Now, I would like to highlight some really important science that I've had the privilege of being, in a way, a bit of a bystander, but endeavoring to contribute in a small way. Sidney Ringer, who is the inventor of Ringer's solution that Hartman adapted to be Ringer's lactate. He also discovered the critical importance of extracellular calcium in exposing frog hearts to solutions where contraction would not occur if there's no calcium in the external medium. And this is really important for the pancreas as well. And George Pollard, with his Nobel Prize on the structure and function of the cell, undertook the work in which he isolated for the first time mitochondria, endoplasmic reticular, and ribosomes, and also identified the way protein is synthesized and secreted in the pancreatic acinar cell. Founder of cell biology, which enabled calcium to become a really important signaling message that we can understand with multiple channels, multiple pumps, and multiple mechanisms throughout the animal and plant kingdom. So it's very ancient. Ole Peterson, who is in Liverpool and whom I was able to join in his lab, made some absolutely crucial contributions to understanding the physiology and pathology of the pancreas. And Ken, He's one of the founders of the company Calcimedica, of more which later. So Peter Heggie, who's unfortunately had to leave, actually drew attention to the pleasure he gets from reading old papers. I'm not sure I get quite the same from reading our own papers, but this is 25 years ago. The key component on this slide is the bottom right panel, where you can see that there is a spike, which is calcium in response to hyperstimulation, but then it returns to the baseline. And the darker line, which in some other plots above it is going up, representing trypsinogen activation, identified by benzamidine, does not occur. And that's because there's no calcium in the external medium. So this is really important. And you can see the hyperstimulated pancreatic acidocyl is all vacuolized in the middle on the left. And to its right is the similar hyperstimulation with no calcium in the external medium. So something about external calcium and its entry. So this really presented us with an opportunity to target lots of mechanisms. And you saw on the previous slide a whole list of papers, and that's all the sort of the work we've done to try to substantiate this finding and identify whether or not it's the case that this is actually acting in all the different forms of pancreatitis. And here we just didn't know what the store-operated channel was. And that's the channel that when the endoplasmic reticulum is emptied, calcium comes in through the plasma membrane. There's another really, really critical component, which you'll all know, and that's the immune activation, the immune amplification, without which pancreatitis would not be what it is. A terrible, devastating disease in some cases. So, great, 2006, along comes Stefan Fesky and his co-workers, and they identify that ORI1 is the pore of this calcium release activated calcium channel entry mechanism. And on the left, is that right? Yeah, it's on the left. Thank you. It's just confusing because you got it around the back of your head. So that shows the overall cartoon of what's going on with ORI and the other molecule, STIM, which is on the endoplasmic reticulum membrane. And when the calcium concentration in the endoplasmic reticulum goes down because of release, STIM's EF hands in the ER join, and the STIM ORI activating region, SOR, is then opened up, exposed and joins to ORI, which then can open as a calcium channel, highly selective hexamer. So Stefan actually, as well as Ken, who I referred to earlier, Ken Staudeman, both founders of Calcimedica in 2006. So they really got at it immediately. And having identified ORI and STIM as this mechanism in severe combined immune deficiency, because that enabled cloning of the ORI channel where it's mutated, that's the immune consequence. Clearly, it's very important. Here, we see the impact of compounds that inhibit the ORI channel. And these are just general parameters of experimental acute pancreatitis, happens to be in the mouse, wherein the compound has been administered either 1 or 6 hours after either bile acid or fatty acid ethyl ester induced acute pancreatitis. And clearly the 1 hour administration works better. But nevertheless, for clinical practice, it's important we have a little bit of time, and the 6 hours does work. So Richard Waldron and Steve Pandol and their group then extended those findings to confirm that the immune inhibition is a contributing factor, the respiratory bursts in neutrophils in particular, and cytokine release, both being significantly inhibited to reduce the severity of acute pancreatitis. And then Li Wen who was a PhD student with me in Liverpool, and now professor at Peking Union. Some excellent work coming out of the laboratory. Developed pancreas and neutrophil-specific ORI knockouts. And in the pancreas-specific ORI knockout, the lung injury was much less, but the pancreas injury was more. And in the neutrophil-specific knockout, data on which is displayed on the right, the pancreas injury was less, but the lung injury was more, demonstrating that we have two mechanisms. We've got the pancreas parenchymal injury from ORI excessive entry, and we have the immune activation injury throughout the body, as well as in the pancreas, making the whole thing worse. So I think it's really important for us to work with industry, particularly with pancreatitis, where there are no drugs at all, because if we don't collaborate with them really, really effectively, the drugs won't be developed. And it's really tough. There are so many hurdles and there's so many potential failures, but this is a great story. So the phase 2b trial of Oxora, which is the only calcium release activated calcium channel drug still in development. There was GSK 7975A, which we worked on. There was Rhizane's compounds, which they're no longer working on. There were Cinta's compounds, which they're no longer working on. But here we can see the impact on time to solid food tolerance is increased markedly. Now, the problem with this trial is it was open label and therefore is clearly open to bias. So then there was a design for a phase 2b with extensive consultation advice taken from the FDA, comparing placebo, 0.5 milligrams per kilogram, 1 milligram per kilogram, and 2 milligram per kilogram zegocractin, which is the approved name. Oxora is a proprietary name. For this compound, CM128, which you saw data on, and CM4620, which is the same compound. So the primary outcome was time to solid food tolerance, and that was predicated partly on the basis of the phase 2a and partly on the basis of results from the Dutch pancreatitis study group. But unfortunately, it did not show an effect across the whole modified intention to treat trial group. However, pre-specified analyses in the statistical analysis plan, finalized before any data were available, demonstrated that there were impacts on patients presenting with a high hematocrit, and patients presenting with Balthazar D or E, that's high scores on their CT. Now, general multiple comparisons and modeling is a particular statistical approach that endeavors to define the relationship between a drug at various doses and clinical impact on a specific measure. And it's widely used in drug development, but it is a fairly niche technique. Nevertheless, it's relatively easy to understand when represented graphically, and you can simply see the relationship between the likelihood that the time is improved with the higher doses as you move across this slide. Perhaps more importantly, although as a secondary outcome, was the impact on severe respiratory failure. That is respiratory failure that requires invasive or non-invasive high-flow nasal cannula ventilation. 8 patients in 95 developing this in the placebo and low-dose groups, whereas none in the high-dose groups. So the modeling that I referred to earlier simply confirms this and validates the finding. So some of you may be familiar with this win ratio. I did bring this into a presentation last year, where one compares one patient in one group with every other patient in the other group and move through all the patients in the first group, undertaking the same comparison, ending up with wins or losses on particularly important clinical outcomes. And they're usually stratified in order of their priority. So clearly mortality has to come first and new onset severe respiratory failure second, and then pancreatic necrosis and length of stay. And when one does that, the wins and the losses for those patients that actually there are wins and losses are taken out, so that as you move down, you're using the subsequent outcome measures to evaluate the remaining patients where there were ties. And that, for the comparison of placebo versus high-dose Auxora, zegocractin, demonstrated the impact overall. Now, this was an exploratory outcome, so it's not really one that you can say, right, well, this is really hard line. But the trial was designed to really achieve three things. One, can we identify patients that we really need to target in the phase 3? And those are patients who are most at risk of organ failure. That's clear from these data. Secondly, can we identify a dose from the doses tested that is preferable to test in a phase 3? Yes, 2 milligrams per kilogram, because that shows the biggest effect. 3, can we identify an outcome measure to use as the primary outcome measure? Because we've found that this time to solid food is not really sufficiently sensitive. And also, although it was read out in all the patients across the whole trial, it wasn't picking up the impact of the treatment. Yes, the win ratio as that. So it really has been a very useful experience. Now, ORI-1 and ORI-2 and 3, but ORI-1 being actually the principal, is distributed widely. It's an ancient channel, Drosophila characterization, structural biology, etc. It's widely dispersed throughout the body on many cell types, but it's doing different things. In the pancreas, it's critical to secretion, but in other places, it's more of a modulator. So would there have been adverse reactions? Sorry. In terms of treatment-related adverse reactions, actually it was slightly more in the placebo group. So we're not getting a readout from this relatively small number of patients, 216, and the Cardia trial, which was in COVID, which couldn't progress because actually by the time it was complete and showing effect, the COVID pandemic was ended. But the safety data are good. So where are we? We know that calcium is a critical signaling messenger in the pancreas. And we also know that calcium overload is common to the vast majority of forms of forms of acute pancreatitis. We also know that the ORI channel is a critical mechanism. It's the only mechanism of calcium entry in the pancreas, but it is probably the most important and leading one. And targeting it in these trials, you can see, has considerable potential in treatment of the disease. So we need a phase 3, and the FDA want it to happen. They actually want it to happen. Thank you. Thanks, Nathan.
Wonderful. And we're going to save question and answer for afterwards. Time next, Professor Aleuch, who's needs no introduction either as a professor of pediatrics at University of Iowa and as one of the world's leaders in pediatric pancreatitis research.
Thank you. Good morning, everyone, and thanks for sticking for the last day of APA. And thank you, APA, for inviting me to talk about pediatric pancreatitis. My job is just to talk about how the disease progresses. I have no relevant disclosures. The goals and objectives of my talk is to give you a brief understanding of the epidemiology and the progression of pancreatitis focusing on pediatrics and highlight the unique features of pediatric pancreatitis and the challenges that we face, and emphasize the importance of early recognition and review potential emerging therapies, which we don't have many. So I will just start by highlighting this paper. This is a paper from China, just focusing on the epidemiology, the incidence, and the burden of pancreatitis currently between the birth and the 24 years of age. What's also interesting that it also forecasted up to the year 2040. As you can see here on the slide, there has been an increase in the incidence of pediatric pancreatitis beginning the year 2000, so the turn of the millennium. This is something Dr. Lowe talked about in his memorial lecture, Frank Brooks' memorial lecture. This is not only the increased incidence, but the pediatricians just understanding that there is something called pediatric pancreatitis, that we understand that this disease exists. But what happened is the incidence leveled off in North America and other developed parts of the world. This paper talked about overall increased incidence across the world. So this continues to be a problem across the world, unfortunately. So you see the burden. The burden overall decreased, I think, because we pediatricians understand how to treat this disease better. But the burden in young kids still very significant. So it's a significant problem for us to understand. This is a patient of mine. I just wanted to highlight the importance of pediatric pancreatitis and how it progresses over time, and as the kids age, to kind of just carry the message across. So this kid presented, like a lot of kids present, with acute pancreatitis around preteen age, 9 years old. So the typical presentation: nausea, vomiting, abdominal pain. The diagnosis was probably not made initially because this kid had recurrent bouts of abdominal pain. But this time amylase and lipase was done, and it was elevated, and the diagnosis of acute pancreatitis was done. No causes was found, but the kid got better. So this was treated, the kid got better, and so this was probably an idiopathic acute pancreatitis, which we see very commonly in pediatrics. And as you can see here, the etiologies in pediatrics is pretty broad compared to adults. In adults, we have mostly alcohol-related gallstones, or idiopathic is a smaller portion, versus in pediatrics, we still have a significant portion of idiopathic acute pancreatitis, but we have a variety of etiologies that we have to rule out, as you can see here. Probably the idiopathic is going to shrink over time as we understand the causes of pancreatitis a little bit better. So what happened to this patient? Like a lot of the kids we deal with, continue to have recurrent attacks of pancreatitis, and diagnosed with acute recurrent pancreatitis at 11 years old. That was probably the time I started following him, and he had the genetic testing. At that time, we were doing 4 gene testing, which is CFTR, SPINK1, PRSS1, and CTRC. So he was heterozygous for R117H, which is in general is a variant of varying clinical consequences. He was a heterozygous, and SPINK1 N34S, also heterozygous, and all those other variants were either negative or insignificant. So when you have those 2 variants, there's an increased risk for acute recurrent and chronic pancreatitis, but for which we don't have any treatment for. So he did not have CF. The sweat chloride was normal. Imaging was normal. So you have a kid with acute recurrent pancreatitis, for which, of course, we don't have any treatment for. He continued to have attacks, and he progressed into the adulthood. So he came back when he was 19 with a very severe acute pancreatitis attack. He was in the PICU, sick for a while, stayed in the hospital for 11 days. At that point, he denied that he was drinking or smoking, even though I just told him repeatedly that those are some of the adult behaviors that he shouldn't be acquiring. But I mean, anyway, when I asked him, he denied those things. Just to review the causes of acute recurrent and chronic pediatric causes in the pediatric cohort, this is from the INSPIRE-2 study. Most of them are genetic. There's a portion that are anatomical. Most of them in the anatomical category would be pancreas divisum. PRSS1 is responsible for acute recurrent to chronic progression, and the CFTR is mostly acute recurrent. And all the other things like environmental etiologies, autoimmune, it's not something we commonly see. And another thing that majority of the kids who progress from acute recurrent to chronic pancreatitis or who have a chronic pancreatitis etiology report multiple attacks of acute recurrent pancreatitis, which means we have a window of opportunity for interventions if we have treatments, but we don't. So, and when we compare, and this is comparing NAPS2 to INSPIRE-2 cohort, so kids mostly genetic, some obstructive or anatomical, versus NAPS2 is mostly environmental. So as he progresses, will that be more like what we will see? I don't know. But I mean, this is what we see. So this kid that I I continue to follow as he became more like an adult. Now he started having more episodic pain. And some of his symptoms were dyspeptic and GERD type symptoms, and we treated those. And I had an imaging study, and this is his MRI. As you can see, he started demonstrating some chronicity. This time, he had a long common channel in his MRI and parenchymal T1 hypointensity, PD dilation, beating and stricturing, dilated ductal side branches. When we had the secretin given to him, I mean, he had very good amount of secretion, so he was not exocrine pancreatic insufficient, but demonstrated chronicity after 15 years of his first attack. So he had an ERCP with a sphincterotomy, and the PD stent was put in, but that didn't stop him from having attacks, which is something we commonly see also. So the progression is acute to acute recurrent, which is about 15 to 30%. So for him, it was 100%, obviously. The genetics play a role. From acute recurrent to chronic pancreatitis, again, genetics play a role. The incidence is about 2 in 100,000. In my clinic, is also, it's pretty common, obviously. Who progresses in the INSPIRE cohort that I'm very intimately involved in? Over 2 years, we have seen 11.5% transition from acute recurrent to chronic, and the average age was 10 to 11. And if I compare him to the others, actually it was a relatively slower progression because PRSS1 patients with obstructive risk factors, and recurrent attacks of pancreatitis actually are the ones who progress faster. So this paper is not published. So how about other genetics? This also is an unpublished data. So we see almost a longitudinal time to progression in the pediatric cohort. If you have genetic mutations, yes, that increases your risk. If you have 2 or more mutations, that also increases your risk. Over time, we started to do deeper genetics. So how about some of the risk factors that you acquire as you become an adult? So this was a paper that we have done with Dr. Yadav and compared the INSPIRE cohorts to the NAPS2 cohort. So what we took in the INSPIRE cohort, the patients who were already over 18, and in the NAPS2 patients who were diagnosed is in the childhood and in the NAPS2 cohort. So when you compare them, they look pretty much the same, almost the same in terms of risk factors. What was different just slightly was the alcohol intake. So probably our patients, as we watch them, they acquire some of the environmental risk factors that we don't want them to take. So the pain patterns, I think the reason I put this in here is the opioid use is not minimal in the pediatric cohort. I think over time we have seen a decline because of the opioid epidemic. This is something we try to emphasize. It was 17% in our initial cohort. The other thing I wanted to emphasize is the pain can be quite variable, anywhere from no pain to episodic pain to chronic pain. But if the pain is chronic, there's a significant association with opioid use. Some of the, again, unpublished data, does ERCP help? We had a lot of patients who underwent ERCP in the INSPIRE 2 cohort. We just wanted to just look at our experience. If there was a pancreatic duct obstruction with stones, there was a response for about 3 years. We didn't have enough data to see if that was sustained longer, but perhaps at least for some of those patients initially. It doesn't apply to my patients. So CFTR modulators, unfortunately, they are only indicated for patients with cystic fibrosis who have CFTR mutations that the CFTR modulators could be actionable on. So we had some patients in the INSPIRE 2 cohort who were treated with CFTR modulators, and interestingly, some of them did not have CF. So this was all provider dependent and site dependent. So if you can see on this slide that the patients with the CF responded, as well as the patients without CF responded in terms of diminished number of attacks. So just putting it together, how my patient would have responded, would he have responded? I mean, he has R117H mutation, het. He doesn't have 5T mutation, which could have been significant. And he has 70 and 90, which are not significant. I found only one patient who had decreased, which could perhaps be similar to him, but that had a 5T variant. So perhaps, but was not available at the time this patient long transitioned to the adult cohort. Pancreatic enzymes, we started treating patients with pancreatic enzymes who are acute recurrent pancreatitis or CP with pancreatic sufficient phenotype, and have seen a decrease in the number of attacks. Mechanism unknown, but this is because we don't have a lot of treatments to offer. And for unknown reason, the patients with SPINK1 mutation in the INSPIRE 2 cohort had a response. So we have a clinical trial planned. Unfortunately, with the government closed, we can't move forward with that. Hopefully open we can, but maybe. So perhaps he could have been a candidate. So summary, acute pancreatitis to chronic pancreatitis is a disease continuum. The burden is pretty high. Patients with acute recurrent and chronic pancreatitis can have multiple attacks. The genetic and anatomical etiologies play a role in disease progression. Most of those kids, before they have chronic pancreatitis, have recurrent attacks of pancreatitis. So that could potentially be a point of intervention. Unfortunately, we don't have treatments. TPIAT right now seems to be the only treatment that provides pain relief and improve quality of life. It's also a major surgery. There is a risk for insulin dependence. Some treatments that I have shown you show promise, but we still have many ways to go before they become a treatment option. We don't have a treatment to address the root cause. We have to address the CP in the pediatric age group, because I just hopefully gave the message to you that they become adults and they will have even more burden moving into the adult cohort. So we need more research. I think pediatric specific trials in patient-centered care, biomarker development, and multi-center collaborations. So I think I'm right on time. All right. Would like to thank my INSPIRE cohort and the consortium and everyone for listening on the last day. Thank you.
All right, it's my honor to introduce next speaker. This is Rip Jyoti Talukdar. He's a professor at the Asian Institute of Gastroenterology in Hyderabad, and he will be continuing this session. Thanks.
Thanks, Phil. And at the outset, let me thank Professor Saluja Belin and the APA for having me here. So my job today would be to speak on the pain phenotyping in chronic pancreatitis, connecting the dots to mechanism. And I think the theme of today's session is what happened over the last 1 year, but I'll extend it a little bit more to put the whole story into perspective. So let's go back to the clinic. So I have two patients here, one a 46-year-old male, the other 38 years old, one having a 3 years disease duration, florid chronic pancreatitis, the first one with a duct stone in the head, dilated duct, the second one with a panductal stone, active smoker, the other is no smoking or any substance use, one a diabetic, the other a non-diabetic, and of course both had a recurrent abdominal pain. And these are the images, or these are the patients where we have that action bias, urge to do something to remove the stone to relief of pain, and both underwent some kind of treatment. The first one was endoscopic treatment as well, followed by ERCP, and the second one had a LPJ. And the results, the first one was pain-free for 8 years, and the second had a pain relapse, the one with LPJ after 4. So I'm not comparing surgery and not trying to say that one is better than the other. But if we look at the real-life clinical outcomes, and many of you who see patients with chronic pancreatitis have, I'm sure, come across this kind of outcome. It's a wide range. It begins from complete sustained relief, like the first patient of ours. It may be initial relief with late relapse, initial relief with early relapse. Some do not have any improvement, and some actually worsening of pain, as the patient we heard just a few minutes back by Professor Ali. And if we look at the actual clinical trials, the sham controlled or placebo trials, all of these trials, the recently published shock trial from our group, another unpublished trial from our group on pancreatic enzymes, the tactic study led by Phil, and the pregabalin study by Soren. All of these, if you see, had a good clinical response, but there was also a significant 40 to 70% placebo response. So why is it so? And that's what we are trying to look at today. So let me take you through quickly through the natural history. So these are not watertight compartments. You can broadly divide the disease into early stage, intermediate stage, where the morphological changes sets in, and late stage of exocrine endocrine insufficiency. But mind you, this is not a watertight compartment. There's substantial overlap. There will be acute exacerbations, and with time, the type of pain would change. And then what happens here now, when we talk about the early stage, there is this little thing called pancreatic stellate cell, and we have the person who discovered this cell, Professor Apte, here today. So what happens during the first attack of pancreatitis, whatever be the etiology, alcohol, gallstones, genetic, there will be activation of these stellate cells. And once this activation happens during the first or second attack, most likely this stellate cell comes back to quiescent stage, apoptosis, or reverts back to quiescent stage. But as recurrent attacks keeps on coming, it may be clinical, it may be subclinical, the stellate cell will start its autocrine and paracrine activation, and there will be a stage of no return, where it will start laying down the progressive fibrosis characteristic of chronic pancreatitis. There will be lots of oxidative stress, and that leads to persistent inflammation. And the problem is we do not know at what stage this stellate cell goes from reversible stage to a permanently irreversible stage. And then this was something described by Professor Zoen Braganza several years back, the concept of oxidative stress and the methionine cycle. So what happens in this inflammation, recurrent inflammation, there will be alteration of the methionine cycle. The methionine is depleted because of the antioxidant defenses, as a result of which there will be loss of exocytosis. And that may lead to small quanta of subclinical or maybe clinical attacks of acute pancreatitis because of loss of exocytosis, pancreastasis, mitochondrial injury, and ER stress. And that, of course, we all know leads to inflammation. Now, what about when the disease progresses to the intermediate stage, when the stones form, the ductal obstruction happens? So there's a beautiful study, it was published long back, where stellate cells were isolated, and in a Petri dish, increased pressure was placed from 0 to 100. But 80 millimeter of mercury pressure, there was significant maximal reduction of superoxide dismutase, which is antioxidant defenses. And when the pressure was released or removed, then there was restoration of this superoxide dismutase, which means that with time, with pressure, there could be more and more stellate cell activation, and that leads to oxidative stress. Now, if we put this into the clinical perspective, just take a patient like this who had a ductal obstruction with an obstructive ductal stone, supposedly having ductal hypertension. So these patients will likely be having activated stellate cell that leads to oxidative stress. And of course, that leads to inflammation. Right? So whatever be the etiology, there will be activation of stellate cells early on in the disease, which keeps on progressing and goes to irreversible state that leads to, the body has a huge reserve of antioxidant defenses, but there will be a certain amount of time, point of time, where there will be an imbalance and there will be pro-oxidant stage that will lead to immune activation, cytokine expression, and inflammation. And that inflammatory mediators would finally lead to the activation of TRPV-PAR1 receptors in the pancreatic acinar cells and the nerves. And this is nociception. This is when the patient starts getting these recurrent episodes of pain, which we call nociception. Now, this is a kind of physiological event. So whenever there is any kind of pain impulse, it would be transmitted to the dorsal horn ganglia, and then it would be transmitted to the brain. And there's a beautiful mechanism in the brain called descending inhibition, which should try to inhibit the outgoing impulses. But what happens in chronic pancreatitis, because of the recurrent inflammation, there will be lots of mast cell activation within the pancreatic acinar cells. And mind you, opiates can activate mast cells. So that's why probably opiates in the long run can cause more harm than good. And then there will be neural inflammation, thickening of the perineural sheath. There will be GAP4-43 expression, a marker of neuroplasticity. There will be a change in the autonomic nerves. And there will be expression of neural chemokines like fractal kite. And these perpetuates the pain more and more as the disease progresses. So what happens when this nociception and neural inflammation begins? So initially, with the acute episodes, there will be activation of the spinal cord of the pancreatic segment, the T10, L12, that area, which the celiac ganglion supplies. But as neural inflammation continues, as the neural transmission continues and the nerves gets sensitized, then things like spinal hypersensitivity happens. Things like inflammatory hyperalgesia, little bit of inflammation can give a sense of huge amount of pain, mechanical allodynia. Even the normal postprandial enzyme secretion would give a sense of pain to the patient. And then that leads to involvement of more spinal segments above and below the pancreatic segment. And this is a stage where if you do a celiac block, the patient is not going to work. So it's a function of time in which more and more spinal segments are involved. And this is what we call the peripheral or neuroplasticity. And then the clinical surrogate of this, the patient will start complaining about more areas of pain, more areas of referral, wider areas of pain, which was beautifully shown in a gastro paper way back in 2007. And this is something that we can use as clinical surrogates of neural sensitization in the bedside. Now, what happens in the brain once the spinal events happens? Now, when there are acute stimuli, those acute episodes of pain, so that is nociception, right? So there will be acute stimuli, the pain will be transient, and the pain has a sensory feature. The patient will be able to talk about the quality of pain, burning, stabbing, pressure, whatever, the location, the duration. But as this sensitization progresses, as the pain transmission progresses, many more areas like the nuclear accumbens, the hippocampus, the hypothalamus also gets activated. And these are the areas which are responsible for effective and contextual aspects of pain. And this is where chronification begins. The pain memory comes in, emotional responses comes in, motivational responses come in. So at a certain point of time, the patient will not bother about the nociception. Now the disease gets converted from a nociceptive disorder to disorder of pain perception and pain experience. So it becomes a disease of the brain, no longer in the pancreas. And this is when no kind of intervention that I showed in the trial, the shock trial, whatever be the trial, the tic-tac, the responses may not be adequate. Right. So, and then the other important aspect I was talking about, descending inhibition, and this is basically physiologically a protective mechanism. But what happens in acute chronic pancreatitis, this descending inhibition is largely mediated by the periaqueductal gray matter, which has the neurotransmitters dopamine, GABA, norepinephrine, glycine, enkephalin. So these mediates descending inhibition. But when there is chronic pancreatitis and recurrent inflammation and recurrent neural transmission, the serotonin in the medial medulla, they have differential expression of the receptors. So if the pink receptor, the HD1A, 1B, 1D, and 7 gets activated, they inhibit descending inhibition. On the other hand, if the 2A and 3 are activated, they facilitate descending inhibition. So this is very important, and it's very difficult to figure out which patients actually have these pink receptors activated and which have activation of the green receptor. So these are the clinical problems that precludes us from using the right kind of treatment in the right situation. And now something more happens. So that was about the nerves and the neural receptors, but what about the psychological features? So in this study, we could show that a substantial proportion of patients, almost half of the patients, have some kind of depression. And similar studies have been shown elegantly by Anna's group and several others worldwide. But what was more important is what we could show as a change in the neurotransmitters in the brain. Especially the hippocampus using magnetic resonance spectroscopy. And if you see here in the left hippocampus, there was a significant reduction of a specific metabolite called N-acetyl aspartate. And then when we looked at the correlation with the different types of depression and severity of pain, so there was a significant negative correlation of pain with the degree of depression and the severity of pain, which means that there is a relationship between the pain, the depression, which could be mediated by the brain metabolites. And this could be a target area for treatment. If we can somehow modulate the metabolites, possibly we can reduce the pain and the correlational depression. So there is a significant negative correlation of the hippocampus N-acetyl aspartate with emotional and cognitive function as the depression and severity of pain increased. So now if we look at this one, this is the traditional way of thinking, the linear way of thinking. If there is a change in pancreatic morphology, some inflammation in the pancreatic duct, some stone in the pancreatic, we have that tendency to break the stone, remove the stones, break it, crush it, or do surgery. But now we know that there are several other factors. There are things like changes in the metabolites in the brain, and there will be lots of cognitive changes, altered brain metabolites, sensitization. So this is a complex cocktail. So that linearity is no longer there. So these are all non-linear, and this is where we face the problem. There will be lots of overlap between the different mechanisms, and it's very difficult to tease out a particular patient at a certain time point which mechanism is profound in them. And that is why probably all these clinical trials do not or fail to show a prolonged and sustained response. I talked about this conversion from nociception to pain perception, and this is where subjectivity comes in. So many of these patients have a subjective response, and it's difficult to tease out which is a real response or which is a placebo response. And now let's look at 3 real-life pain phenotype. So this is patient A, who had the pain for around 3 months, and then there was no pain for at least 12 months. Probably the pain-free period may extend longer if we follow the patient. Another patient who had pain for 3 months, there was a pain-free period for around 3 months. Again, he had pain for 3 to 4 months. And the patient had a short pain period, and then again there was a prolonged pain period at a pain-free period. And the mechanisms may be different. Now let's put them in clinical trials. So all of them would fulfill the criteria of clinical trials in chronic pancreatitis, usually 3 episodes of pain for past 3 months, the definition of chronic pain. And this is where the enrollment happens, and this is where the endpoint, just arbitrary, right? Now the first patient, if the patient was treated with an active drug or intervention, he by natural history, he will not have pain, but it will give a false impression of the improvement of pain because of the intervention. If the patient is in the placebo group, again, there will be no pain because of the natural history. Don't do anything, he will not get any pain, but that may give a false perception of a placebo effect. In the second diagram, second patient, initially possibly the first pain was of a different mechanism because of probably ductal hypertension, but later on there may be a change in the mechanism because this mechanism can keep on changing based on the risk factors. So because of that, the patient may have a relapse, and we may perceive there is a failed treatment, but the problem was because of a new mechanism. And the third, maybe the patient from beginning had a neuropathic pain, but we are treating them with a SWL or some surgery. So we are treating a wrong target, and therefore this patient may not respond. So this makes the whole thing very heterogeneous. And that is where pain phenotyping becomes very important. So how to do this? Firstly, I had already talked about the different types of pain character as the disease progresses. So this could be simple clinical surrogates where at the bedside, you can just think by a good history, this change in pain pattern, character, the triggers, involvement of new areas. These are soft markers that can make us think possibly the patient has some kind of sensitization going on. And then the pain consortium that we are working on, many of you in the group maybe, I think, are members here. So one thing that we are doing is to identify quantitative sensory testing. Several studies, I won't go into the details of those studies, but several studies have shown the benefit of pancreatic quantitative sensory testing. Then to look at the ECG and EEG variability. That's me, but I was not the patient. I happened to be the first healthy control in the EEG study. So that's how I'm there. So again, the variability of the changes in EEG and ECG variability could be markers. And then there are several patient-reported outcome measures, the PROMs that we are trying to evaluate. One of them is the COMPET SF, and validity has been shown in different languages, things like hospital, anxiety, depression, pain catastrophizing. So these are some of the features or some of the tools that can be used to do a pain phenotyping. But the problem is the geographic variability. When we looked at this in different parts of the world, from in India, in US, and Europe, there has been geographical variability, probably related to the sociocultural and many other factors. So this is where we need to harmonize the data, and probably that's going to be the future. So the last slide here. So if we put all together, this was the natural history that traditionally believed or understood, but now we have this concept of pain sensitization and anxiety and depression, but we don't know when sensitization sets in. And that is why it's very difficult to select out a patient for a specific type of treatment. But to put the overall thing into perspective, possibly antioxidants will work, and we have shown in our clinical trials to work if we start early on, especially for non-smokers, non-alcoholic, and those not on opiates. The intervention can work. I think there is a very small window where the intervention will work. And this is where the pain phenotyping becomes important. And these are the patient who has neuropathy and non-neuropathy. The neuropathic pain, of course, goes to neuromodulators, and the other group may be benefited by interventions. And finally, I think we should look at pain in CP through a new lens. We have to understand a new concepts. Think beyond the pancreatic duct. It's not the duct that is disease. That whatever we see in the duct is a complication of the disease. So treat the acinar cell, treat the inflammatory cells. Pain phenotyping important. And I think pancreatic stellate cells should be the early target. Large number of animal studies have shown benefit of targeting pancreatic stellate cell on the disease, the reversal. But I think now it's time to look for new molecules. The brain and nerves should be the new targets. And I think it's time to redefine clinical trial designs and outcomes. So I think I'll stop here. And thanks, and happy to take questions.
All right, the last speaker of the session, it's my honor to introduce Shaunak Majumdar, Associate Professor at Mayo Clinic in Rochester, and he'll be talking to us about fatty pancreas.
Good morning, everyone, and thank you, Phil. I really appreciate the.
Can we reset the speaker's clock to 15 minutes? Thanks.
No worries. I really appreciate the extra time I was getting and the APA leadership for selecting this topic of fatty pancreas. You know, over the years, we have all come to realize the impact of obesity on human health and disease. And if we look at the field of fatty liver disease, there is so much that we know today about the link between liver fat and fibrosis and inflammation and carcinogenesis. And if we turn our attention to the pancreas, I think it's fair to say we know very little about how pancreatic fat affects all of those conditions. And looking at this very broadly, I think one of the limitations in the field of fatty pancreas disease is our inability to rigorously and objectively quantify pancreatic fat. So over the course of the next 15 minutes, I'll try to share with you what we know about the clinical relevance of pancreatic fat, what are the limitations in our current approaches to how we measure it, and how we can overcome that and hopefully move the field forward. So broadly speaking, you'll see many terms used to describe fatty pancreas in the literature, but they can be broadly categorized into fat infiltration and fatty replacement. Fat infiltration is what we would consider in the setting of adiposity and obesity, and fat replacement would be where there is a sinner injury and cell death, and the fat moves in to fill the empty space. And my journey looking into fatty pancreas started in the clinic, like many other research ideas that we work on as physician scientists. When a relatively young individual with an extensive family history of pancreatitis and pancreatic cancer had imaging done somewhere else that showed a fatty pancreas and came to us asking whether she should be concerned. And we really didn't know the answer, so we looked into the literature, we wrote this paper and asked the question, should we be concerned? At the time, this is almost 6-7 years back now, we came up with two questions. We really did not have a good understanding of what the clinical relevance is, and there was no way to objectively diagnose fatty pancreas. And over the course of the past few years, there's been multiple studies now adding to our knowledge of what the clinical relevance is. It's a very nice study done in a cohort in Hong Kong that showed that pancreatic fat is associated with a higher incidence of diabetes mellitus, and for every 1% increase in pancreatic fat, the risk of diabetes increases by 7%. And here we are talking about true incidence of disease, not merely associations, and adjusted for other factors such as obesity and fatty liver disease. Pancreatic surgeons, if any are in the room, will tell you that they do not like increased pancreatic fat because there's a risk of increased postoperative pancreatic fistulas after resection. And work done by Vijay Singh and others have shown very nicely that acute pancreatitis is worsened significantly in the presence of pancreatic fat due to lipotoxicity. This is recently published work by one of my former fellows, Arjun Chatterjee, who's now at Cleveland Clinic, showing that over a median follow-up of about 4 years, if you compare patients with and without increased pancreatic fat, there appears to be a signal for higher incidence of both neoplastic and inflammatory pancreatic diseases, albeit the sample sizes are small, and it's challenging to make definite conclusions. And this, again, in the same line of understanding clinical relevance, this is from Max Petrov's group, published online in the Red Journal last month, that combines 23 studies looking at what does excessive intrapancreatic fat do to all these diseases. And you can see not only that did they conclude that there is a higher risk, but a significant proportion of patients with these conditions could be avoided if excessive intrapancreatic fat was eliminated. While all of this is adding to our understanding of the clinical relevance, I think the field remains limited by the fact that if you dive into the different studies included in these types of analysis, there is significant heterogeneity in terms of how fat was estimated, what modality was used, where the threshold was drawn for determining high intrapancreatic fat versus normal intrapancreatic fat. And as you can understand, if the objective definition for the entity is not established, then that really jeopardizes the interpretation, especially when it comes to causation. So how do we measure pancreatic fat? There are various imaging modalities that we use to image the pancreas, and obviously histology is always considered the gold standard. Transabdominal ultrasound, unfortunately, is not a great tool for pancreatic imaging. Endoscopic ultrasound, there are studies showing that you can use hyperechogenicity of the parenchyma, obscure duct margin, and blurring of the salt and pepper appearance, which can all be then used to say which gland is fatty versus not. There is inter-observer disagreement sometimes on these metrics, so they remain somewhat subjective. CT can be used for measuring pancreatic fat, but only if it's a non-contrast CT, because the use of contrast really changes the parenchymal intensity and any kind of estimates you can get regarding organ fat. So over the years, MRI has really become the gold standard, not only for measuring liver fat, but also for measuring pancreatic fat, using the same principles of proton density fat fraction. The question that was often asked is, how does that relate to histology? Histology in the pancreas is challenging because biopsying it comes with the risk of pancreatitis, and it's a fairly invasive procedure. So it's challenging to do large-scale studies looking at pancreatic histology and comparing it to fat. But there was this study, small sample size, looking at the correlation between MRI fat estimation and histology on pancreatectomies that showed that there's a strong positive correlation between histologic fat and MR assessment of pancreatic fat. Now, I want you to really pay attention to this figure. This is from a publication from several years ago. And what it's showing here is that if you take one patient and you take 5 different anatomical locations, in this case, the right and left lobe of the liver and 3 different regions of the pancreas, if you were to place regions of interest, random regions of interest, and measure fat content in that region, it really doesn't matter in the liver where these regions are placed. So each dot there is the fat fraction from a random region of interest. So you could place these regions of interest anywhere in the right lobe of the liver. You'd come up with similar proton density fat fractions, but that's not the case in the pancreas. So even in the pancreatic head from a given patient, depending on where you're sampling, the proton density fat fraction can be highly variable. So while we have this gold standard of fat fraction, typically in our workflow, radiologists will tend to use regions of interest. And while that works in the liver, in the pancreas, that's really a problem. And so this is my problem statement. I call the fatty pancreas problem. We know it's associated with pancreatic cancer risk, increased risk of diabetes, increased severity of pancreatitis. We cannot easily biopsy the pancreas, and the fat fraction approach on MRI uses regions of interest that can be limited by heterogeneity. So several years ago, we asked the question, can we do a whole pancreas fat assessment and overcome this problem? And we turned to AI to solve this problem, and I'll share with you a little bit about how we went about it. So we started with CT scans because back in the day, it was relatively easier to segment CT images using AI models. And this is nearly 20,000 images on over 400 CT scans, all non-contrast, that we did segmentation. And then at a whole gland level, we used Hounsfield unit intensity. So not a perfect measure, but we wanted to prove the concept that we can actually do whole gland measurements of radiomics features. And that allowed us to establish over a population of males and females what happens to the Hounsfield unit intensity and identify age and sex-based percentiles of fat across this population. We also looked at pre-diagnostic CTs from patients who developed pancreatic cancer and noted that in the pre-diagnostic phase of pancreatic cancer, the CT whole pancreas Hounsfield unit intensity appears to be lower in patients who then went on to develop pancreatic cancer, adding some indirect evidence to the fact that there might be more pancreatic fat in the pre-diagnostic phase of PDAC. We then translated this into MRI-based measurements. The first question we asked, and this was work done by Lawrence Janssens, who was a former GI fellow at Mayo Clinic, is how does CT estimations of pancreatic fat compare to MRIs? So we took a paired cohort of patients with both CTs and MRIs. And it turns out that there's very strong correlation between these two entities. As the CT Hounsfield unit intensity goes down, the MRI PDFF or fat fraction goes up, which is what we would expect. And in that study, we went back to look at what is this heterogeneity issue, and is that really a big problem? And we concluded that yes, it is. So in individual patients, if you take different axial slices of the pancreas, you end up with very variable fat fractions. So to really have a true estimate of a person at an individual level of their pancreatic fat, I think averaging over the entire pancreas is of critical importance. So I hope I've been able to convey to all of you that pancreatic fat is heterogeneous and we need a whole organ approach for fat measurement. While this is a need, it really is not clinically feasible for radiologists to sit and segment entire pancreases and do these whole gland measurements. And that's where deep learning can be very helpful with these models that take a pancreas MRI, do the segmentation, and give the output as the whole pancreas fat fraction.
And this is work that we continue to do within our team at Mayo, and several other groups around the world are also working on similar ideas. As you can tell, on one side is the patient whose pancreas was segmented and the mask was created by a human radiologist, an expert, and on the other side is the mask created by AI. I'll help you out because without labeling, it's really hard to tell which one was done by the human versus the AI model. And while it's not segmentation of pancreas on MRIs is not perfect, there are imperfections, so we don't get a Dice coefficient of upwards of 9, which is what we sometimes get with CTs. We looked at how does the fat fraction correlate between experts and the AI model. So we had 2 human readers spend a lot of hours really annotating 30 MRI scans, and we compared it to the AI model. And as you can see, the green line, which is what the AI model measured as whole pancreas fat fraction, almost perfectly overlaps with human 1 and human 2. And if we have a tool like this where we can measure the pancreatic fat, what can we do with it? So this is another group that has worked on the UK Biobank that has nearly 30,000 pancreas MRIs. They do not use the ideal sequence, which is what we use for proton density fat fraction, but they had Dixon scans, and this group looked at what happens if we measure whole pancreas fat and follow these patients up, and concluded that there is a 3-fold increased future risk of pancreatic cancer in patients who had more than 10% intrapancreatic fat as a threshold. We in our group have looked at fatty pancreas in individuals at risk for pancreatic cancer. As I shared with you, that fatty pancreas is associated with an increased risk of pancreatic cancer, and we know family history and germline risk is also associated with pancreatic cancer. All our endosonographers and radiologists will tell you that it is really challenging to detect a small pancreatic tumor, a solid cancer, in the midst of a very fatty gland. So we asked the question as to whether these high-risk individuals truly have more pancreatic fat compared to those without these risk factors if we match for age, sex, glycemic status, and BMI. And this was work done by Tom Konikoff, who's a postdoctoral fellow on our research team, showing that there is really no difference if you match for all these covariates. High-risk individuals do not have more pancreatic fat compared to those without these risk factors. And here's where we are headed with this work. Anup Mishra, who's the lead AI scientist on our team, is looking at can we go beyond quantification to where we can actually generate slice-level heat maps to look at spatial distribution of the fat and come up with images like this, which will then allow us to apply image embeddings to study whether the distribution of fat in a particular pancreas is associated with predisposition to certain diseases or outcomes in pancreatic diseases. So there are several advantages to this kind of image-based assessment of pancreatic fat. It's automated, it's objective, it's quantifiable, reproducible. It overcomes the heterogeneity, and we can see correlations with other measures of body composition. What are the limitations? We don't know where the fat came from, so we need to understand the biology of pancreatic fat better. We don't measure the chemical composition, and it can be challenging in cases that have altered pancreatic anatomy. So to summarize, pancreatic fat is associated with a wide range of pancreatic diseases. We need a standardized objective measurement system, and AI can help. We do need large cohort studies to establish what is the normal threshold of fat in a US population. And there's more work that needs to be done on the mechanistic link between this and pancreatic diseases. So with that, I would like to thank all the people who helped do this work. Hiroaki Takahashi, who's a radiologist at Mayo, who's a key collaborator, and Lawrence, Anup, and Tom, and the rest of the team that I have the chance to work with at Mayo. Thank you.
All right, we'd like to invite all of our speakers to come back up to the stage. We have 7 minutes left of this session. We realize this is bumping up against a break. People need to check out, catch flights, etc. But this was a great session. I'm glad that the faithful remnant could stick around for this. Sohail, do you want to open up with questions? And let's try to keep it really focused and brief.
Absolutely.
Not because you have a bad habit, but just General comment.
For both
Nothing
reasons.
against you. Yeah.
I do have a bad habit,
Yeah.
but I'm working on it. And I wanted to ask the question about CFTR. Maybe I'll just keep it to one question. It's so fascinating, the studies that you showed, Aaliyah, where CFTR modulators seem to reduce the recurrence of pancreatitis, acute pancreatitis, and I missed it because there's a lot of data. Was there any difference in the reduction in risk between those who have a CFTR variant and those who do not?
So this is just only looking at 28 patients. So we didn't go back and look to the rest. So this is just reporting on those 28 patients. So we do not have the luxury of comparing how did those other patients have done. This is just looking over time, how did those patients do before and after the modulators. And you can see that there was almost like a night and day. So, I mean, they had multiple attacks of pancreatitis before they were put on and almost just went down to nothing. So it's not a head-to-head comparison, if that answers your question. But we need to do the trial to answer your question, but we don't have the modulators, at least not yet, to do the trial. Thank you.
Thank you to all the speakers for some incredible presentations. My question is actually to Dr. Majumdar about the radiomics with regards to fatty pancreas. So as we know, there is a heterogeneous distribution of fat as you've been displaying within the pancreas. Is your group also factoring in the fact that fat throughout the body is distributed in a heterogeneous manner? Clearly, the liver has a much better capacity to take up fat, and in a condition, say, pancreatitis, that may play an even greater part in increasing the systemic injury or responses, also considering subcutaneous fat and so on. So in development of these models, are you factoring in an assessment of extra pancreatic fat as well when developing the models?
Yeah, thanks for your question. I think that's a very relevant point. We do have a way of doing body composition, both for visceral and subcutaneous fat. Those pipelines are much more established in CT imaging than they are in MR. And as part of an ongoing study, we are looking at, again, patients who have paired CTs and MRIs, if we can pull in the CT body composition into the pancreatic fat model and how that affects outcomes as it relates to certain diseases. So work in progress. Hopefully I'd be able to share some of that in the future.
Great, thank you.
I think this was an amazing session. All four of you did really amazing job. My only thing is that maybe we should have this on Thursday and Friday so that more of us could have benefited from this. So great talks. But Roop Jyoti, you know, obviously your presentation was great. Data is great. I think you should get an award for the best slides for sure in the whole session. So
Thank you so much.
And we'll talk to the Secretary Treasurer about that.
Obviously, I have a little bit selfish interest that Roop Jyoti was part of my group at one time. But my quick question is that, is the mechanisms what you showed for pain in pancreatitis, and we know there's a lot of pain in pancreatic cancer, are these similar mechanisms or they are very different?
Yeah, so in cancer, events happen much faster, and possibly the cancer stellate cells and the cancer cells are also contributing to the mechanism. So it's not exactly the same. The fundamental concepts are same, but it happens much earlier with more contribution to the cancer cells also. That has been shown. But in chronic pancreatitis, it grows over time, initially to the recurrent acute, then there will be sensitization, and then there will be chronification. So it's not exactly the same, but and therefore in cancer patients, the celiac neurolysis kind of treatment works much better because they develop much earlier and you can target them before other sensitization events happen. In contrast, in chronic pancreatitis, Once the sensitization happens, if you remember one of the slides, you chop off the celiac, it doesn't work. And that's what we see in our clinical practice. It doesn't work. So it's a matter of time and the early involvement of the cancer cells in cancer pain.
Thank you. Can I just ask you one more quick question to Shona?
No, we're short on time.
He's kidding.
I was kidding, but I do have we have 1 minute in the session. I'd like to pose a question to Professor Sutton on a different topic. We'll come back.
We'll
1
end
minute.
the session by 5 minutes.
Anything.
All right.
I just have a
Quick.
very naive question for Shunik because I want to understand a little more this area. Do obese people who have more diabetes, more pancreatic cancer, more pancreatitis also have more fatty pancreas?
Yes, the intrapancreatic fat content tracks with BMI. So patients with higher BMI will tend to have more intrapancreatic fat, but the absence of obesity does not rule out excess intrapancreatic fat. So there are patients who are non-obese who will have excess fat in both their liver and their pancreas. And we just need to understand that better.
All right. Sorry to Rich and Minody. As a moderator, I would like to pose the last question of the session to Professor Sutton. So looking back, your first publication on calcium channel signaling was in the 90s when many of us were not even in science. Could you, in 1 minute, share some words of wisdom related to, for inspiration as it relates to the resilience that it takes to get from bench to bedside over 30 years?
Sorry. I don't know that necessarily I have anything remarkable about this other than that I'm a very tenacious person and I like to follow things through. I think that the one thing that was key is that we do not have a drug for acute pancreatitis. And at that point in time, the 90s, it was quite evident that loads of things were being thrown at the pancreas, but they weren't working. And it demonstrated a complete lack of understanding of mechanisms. So we had to go back to the drawing board and look really at the pancreas to try and work out how it works. And that's where Oli came in. And then the discovery of the fact that excess calcium entry was a critical thing sparked a whole line of investigation that needed to substantiate, to confirm, to validate, to strengthen, to extend, to work out implications. And we were fortunate that there was a channel that was clearly identified as being instrumental in 2 systems, not just one. And people talk about needing to target the pancreas in 2 ways to try and get an effective therapy. And it's taken off from there. But then I've been fortunate because I'm a clinician, so I could move immediately to patients. And in the United Kingdom, the majority of patients with acute all pancreatic disease come to surgeons who specialize in them. And so I had it on my doorstep. So I've just been very blessed. Thank you.
Thank you. All right. Thanks everybody for attending the session. We have a 10-minute break and then we'll come back for the grand finale. Thanks.